When you look at an everyday piece of food, what you're seeing is an illusion of solid matter. Zoom in far enough, and that solid shape dissolves into billions of microscopic building blocks. We all know our bodies rely on carbohydrates, proteins, and fats to survive. But identifying these invisible nutrients inside a random substance requires undeniable chemical proof, not guesswork. The core rule of biology is that all living organisms are constructed from a specific set of organic molecules, and every single one of those molecules is built from three primary elements: carbon, hydrogen, and oxygen. To uncover them, we are stepping into a diagnostic lab. We'll use specific chemical reagents that trigger observable, highly reliable color changes to unmask exactly what is hiding inside our food. Mastering these specific tests allows us to reverse engineer living things. We can observe exactly how biology is constructed, molecule by molecule from the ground up. Let's start with our first unknown liquid sample. Our objective here is to test it for simple sugars. We add a blue liquid called Benedict solution to the sample and submerge the test tube in a water bath heated to between 60 and 70 degrees Celsius. Then we wait for a reaction. The liquid doesn't stay blue for long. It shifts to a distinct orange and finally settles on a deep brick red. That brick red color is the definitive signature of reducing sugars. It tells us the sample contains simple, single sugar units, known as monosaccharides, like glucose. Next, we test a solid sample, like a slice of potato. This time, we apply a few drops of an orange-brown liquid called iodine solution. The reaction is immediate. The iodine hits the solid surface and shifts instantly from orange-brown to a dark blue-black. This blue-black result confirms the presence of starch, which is a complex carbohydrate. These complex carbohydrates are simply massive architectures. They are built entirely by linking hundreds of those simple glucose molecules together into long chains, called polysaccharides. Moving down the bench, our next goal is to identify fats and oils, which biologists call lipids. We perform the ethanol emulsion test. First, we mix the sample with ethanol and shake it vigorously. Then we pour that mixture into cold distilled water, and a cloudy, milky emulsion instantly forms. That cloudy layer forms because of the distinct molecular architecture of a lipid. Each molecule consists of one central glycerol unit chemically bonded to three long fatty acid chains. Our third diagnostic check targets proteins. For this, we use biuret solution. When we add the pale blue biuret reagent to a protein sample, the liquid undergoes a dramatic shift, turning entirely purple or violet. Proteins step up in chemical complexity. Using carbon, hydrogen, and oxygen, they introduce a new element, nitrogen, and sometimes sulfur, combining to form an amino acid. There are 20 different types. They link end-to-end into long chains, folding into specific, complex shapes. By simply changing the sequence of those 20 basic amino acid blocks, cells can construct hundreds of thousands of unique proteins, each built to perform a specific job in the body. We have one final diagnostic tool on our lab bench, a test tube filled with a vibrant blue dye called DCPIP. As we add drops of a food solution into the test tube, a striking reaction occurs. The intense blue dye turns completely clear. That rapid disappearing act is the exact chemical proof we need to identify the presence of vitamin C. Through these targeted chemical reactions, we have unmasked the microscopic universe of carbohydrates, lipids, proteins, and vitamins that hide inside the matter we interact with every day. If our physical forms are just vast collections of these raw molecules, something has to provide the instructions to assemble them in the precise order needed to sustain life. To find it, we leave the chemical bench and travel deep inside the nucleus of a cell. Here, we find the ultimate biological blueprint, DNA. Deoxyribonucleic acid, or DNA, is a massive molecule constructed from two long strands wound tightly around each other in a shape known as a double helix. Spanning the gap between those two strands and holding them together are chemical structures called bases. They connect across the center like the rungs of a ladder, bonded together by hydrogen. The genetic code relies on a strict, unbreakable rule for how these bases pair up. Adenine always pairs with thymine, and cytosine always pairs with guanine. An A will only ever lock with a T, and a C with a G. Mastering the chemistry of life provides a complete picture. You now understand the theoretical genetic code that designs living organisms, and you possess the practical laboratory skills to prove those exact building blocks exist in the real world.